Go. Okay, so the first thing we need to do is finish up this chapter 18. Okay, and we're kind of leading into. Sorry, I got Zoom always likes to throw everything around on me here. Okay, all right, we're using multiple monitors. Okay, so it's terrible. Just right there. Finishing up this chapter, it's actually not a I find this to be like a nice little grouping in here. Okay, and we're just breaking stuff down, but this last little section is where it starts to get a little crazy because you can start seeing everything is interconnected. Okay, and I'm going to go with an analogy to that's probably not relatable, but I'm going to do it anyway. I was a big fan of Douglas Adams' Hitchhiker's Guide to the Galaxy series of books when I was a kid. There have been, radio plays, television shows, and even a movie. Actually, maybe two movies. I'm not sure you want to count that one as a movie or not. But most recently, there was one, and then maybe 10 or 15 years ago, a movie adaptation of it. And it's very absurdist. But one of the things that happens to one of the characters in there is he goes into some place and he's supposed to be tortured by some alien race, but they put him in a little machine and it shows him his relation to the universe, right? And because it's all interconnected and it's just way too much for anybody's mind to handle, and everybody goes insane and dies from it. He comes out of the machine, is like, "That's awesome," because he's the center of the universe. Turns out there's an alternate dimension that he's in that somebody created just for him, so he's the most important person in that little universe that he's in there. But the key is everything here is interconnected, and it can start to drive you crazy if you try and memorize everything unless you have you're what was it neurodiverse enough that you have an eidetic memory and just everything fits into place in there. Most people aren't, and I look at even like Nobel winners and they've done some crazy things to visualize what they see in here. So with all that said, I want to make sure that we don't go crazy, and with seeing all the interconnections that are in here. Um, so we're going to try and kind of ease our way into it and just understand what's going on. I want you guys to memorize the TCA cycle, but other than that, there's not a lot that I want you to memorize because this would rapidly get crazy. Okay, so memorizing the TCA cycle is just enough to kind of wet your brain for instant recall of information. But all this we'll just work on as we come through it. Okay, so where we kind of left off last Wednesday was, just kind of summarizing the way amino acids are catabolized within the body. Okay, so this kind of shows what's going on here. We have our, our essential and our non-essential ones, but we're breaking it down in here into our ketogenic in blue, okay, and our glucogenic in orangish. I don't know, whatever color that shows up for you guys as. And you can see that, leucine, lysine, phenylalanine, tryptophan, tyrosine, and threonine to some extent can break down into acetyl-CoA, right? And many of these can go into acetyl-acetyl-CoA, get converted into ketone bodies. Okay, so here we are with our nice little ketone bodies that are coming in. And we can use those for energy that way. The rest of these amino acids will be broken down into either pyruvate, oxyl acetate, fumarate, succinyl-CoA, or alpha ketoglutarate. Okay, so those are all glucogenic, and you can see clearly the ones that break down into pyruvate or oxyl acetate because those can ultimately get converted into glucose. It's a little more obvious there. It's a little bit harder with these over here because they have to go through the TCA cycle to get to oxyl acetate before they can get converted back into glucose. Okay, but these are all glucogenic because they can make glucose and raise glucose concentrations in the bloodstream. If anybody has any experience with diabetic patients, monitoring that glucose level is important. So if you look at the different proteins that you eat, or the patient eats, you may need to keep account of those to make sure that the blood sugar doesn't rise too high. All right. So this is kind of our nice little summary here. So now we're going to step in and take a little more detailed look at some of these breakdowns. Okay. Oops. All right. So if we look at the ketogenic amino acids, all right, awesome. We can see some pathways that happen here. So if we look at tryptophan, we can actually release alanine in here. And if we make alanine, it can get converted to pyruvate. You're like, well, wait a minute, isn't tryptophan going to pyruvate? Not really, because it's releasing the alanine, but we're actually making this ketoadipate in here that's ultimately going to combine with coenzyme A to make our acetoacetyl CoA. Same thing for lysine. We have a bunch of steps in here, but it ultimately gets to this, was it 5 carbon, 6 carbon ketoadipate, okay? And you can see we cleave off a single carbon group as carbon dioxide to get to our glutarol one, and we cleave off another one to get to our 4 carbon acetoacetyl acetate. If we cleave off that aceto group, then we have our acetyl CoA in there, okay? Phenylalanine will be, see we add a hydroxyl group to give us tyrosine in there. We can release fumarate and get acetoacetate, which can be added directly to coenzyme A. Okay? Leucine, you can see we can actually put it into acetoacetate, or we can have it come in here to acetyl CoA. Okay, so you can see basically depending on which carbon atoms go where. So the blue ones go to acetyl CoA, and the red ones can go to acetoacetate, and combining with the carbon dioxide in there. Woo, good stuff, right? But the whole key is for all of these phenylalanine, tyrosine, leucine, tryptophan, and lysine, and isoleucine, can be converted to acetyl CoA. So they are ketogenic. We're making our ketone bodies in here. Okay, and all but isoleucine can be converted into acetoacetyl CoA. And you can see we're leaving out lots of steps in here, right? Bunches and bunches of steps. So there's lots and lots of reactions and enzymes that are involved in this breakdown to transfer stuff around and cleave. And you can see we're making NADH here as we break this down. We're using up coenzyme A in various places in there, and oftentimes releasing carbon dioxide. Sometimes we're actually absorbing carbon dioxide in the case of leucine, but anyway, we can kind of play around with this. Okay, but these are the various pathways, and you can see ultimately we can make pyruvate, fumarate, or our ketone bodies. And you can see here we can make succinyl CoA out of isoleucine as well too. Okay, so let's talk about some of these in detail, because they have all these steps just to break them down. Why do we have so many steps to rearrange stuff? Well, it turns out that when we start degrading amino acids, we can use them as the precursors to build other things. So in the case of tryptophan, we can use the tryptophan ring here. You can see here's our larger ring structure. We can use that to make niacin, okay, which we can ultimately use to make NAD or NADP, okay. We can also take our whole ring structure there and make serotonin. Okay, so here we have our neurotransmitters. We all know about our serotonin. It's like, oh, I feel good, stuff like that. If we are a plant, plants can use tryptophan to make indole acetate, which is a growth factor for plants. So these are 3 really, really different compounds, but they can all be made from a tryptophan. Okay, and it just kind of depends on where those pathways go. So this is the crazy part. Hey, we're just breaking down some nice little proteins. Oh, we got some tryptophan. Eat your Thanksgiving turkey high in tryptophan, and you can make some serotonin to feel good, or you can make some, niacin, or well, you're not a plant, so you can't make indole acetate, but feed your. Never mind. Going a little crazy here. If we look at phenylalanine, okay, so in order to get from phenylalanine to acetoacetyl CoA, we have what 7, 8 steps? 1, 2, 3, 4, 5, 6, 7 steps in there. Okay, 7 different enzymes: phenylalanine hydroxylase, tyrosine. Because first we get our transfer in here and all this stuff. If any one of these goes wrong, we get disease states. Okay, so our first step in the degradation of phenylalanine is we are going to add this hydroxyl group to it. So phenylalanine hydroxylase, adding a hydroxyl group there to make tyrosine. Okay, and you can see that this requires NADH, it requires oxygen, and our cofactor is going to be tetrahydrobiopterin. It's another one of our nice little tethering molecules in there, but a fun little cofactor in there to add that hydroxyl group on there. If there is a defect in phenylalanine hydroxylase, you end up with phenylketonuria, or PKU. Okay, what this does is it leads to a buildup of phenylalanine, which has all sorts of nasty little mental and cognitive effects to it as well too. The treatment is just don't eat a lot of phenylalanine. So have a low phenylalanine diet. Okay, because your body can certainly use some phenylalanine because it's got incorporated into the proteins that it makes, but excess phenylalanine can't be broken down, so it accumulates and has all sorts of nasty effects. Okay, suppose we get the tyrosine. Well, we can transfer an amino group off of there. So here's our aminotransferase. Again, a lot of this chapter is looking at moving around our amino groups here. So tyrosine aminotransferase, if there's a defect in that one, you end up with this tyrosinemia 2 disease. Okay. Once we get to this compound here, well, now we have this dioxygenase. So essentially you can see that we are, you know, what are we doing here? Cleaving off a carbon dioxide and adding an oxygen group over here. Defect in this enzyme gives us tyrosinemia 3. If I jump ahead, we've got a tyrosinemia 1. So again, all this is dealing with tyrosine. The breakdown of tyrosine is kind of where we go from phenylalanine to tyrosine. Okay. We have a couple other intermediates in here. If we have some issue with this particular enzyme, there's this alkaptonuria. Okay. Very similar too. I like this. There's what? No disease state with this one? Okay. No. All right. So apparently, if we build up um, malacetoacetate, malacetoacetate, maybe that's not that bad. I don't know. Maybe there are other ways of getting there too, but or we're just doing too much in here. But these are all different diseases. These are all heritable diseases as well too. So if there is a homozygous, sometimes heterozygous defect in any of these enzymes, offspring can develop these diseases as well too. All right. We will play around later on, probably on Wednesday, with looking at some of these diseases and what they do, how their effects occur. And again, it's just all accumulation. In the case of PKU, it's accumulation of phenylalanine. In the case of tyrosine type 2, it's accumulation of tyrosine. Here we'd have an accumulation of this parahydroxyphenylpyruvate, right? And in here, tyrosinemia 1, it would be an accumulation of fumarole acetoacetate. A lot easier for me to say than mallyl acetoacetate for some reason. But anyway, so all these various enzymes, a ton of enzymes involved in just breaking this stuff down. And if there's a defect in one of them, you can end up with disease states. Some of them are more serious than others. Okay. So let's talk about PKU because it's not horribly uncommon. I mean, it's fairly rare, but it does show up it's much more common than a lot of the other amino acid metabolism diseases in here. But you get a buildup of this phenylalanine and phenylpyruvate because we can substitute. We can basically cleave off the alanine and add a pyruvate on there as well too. But this is working through our aminotransferase again here. But if we have these two compounds in here, these can kind of be broken down into this phenylacetate and phenylactate, which have deleterious effects on neural development. So we basically end up getting people who are mentally challenged. Basically, there's a delay or a decrease in the development of the intellect and in neural development in there. And again, if you just eat a regular diet, then you're going to have a child who is intellectually in intellectual decline and far behind students and won't thrive and will have issues. However, if you limit the intake of phenylalanine, these children can actually have really normal lives. You just have to watch what you take. I don't know if any of you have noticed, but there are a lot of nutrition labels I'm old. I've seen the development of nutrition labels. It used to be like, oh, there's nothing on there. Oh, it's like now it's just how many calories in there, and then it's how many calories, how many sugars, all that. And then you'll see like, well, is this glutagenic or ketogenic? Or a lot of times they'll say, is there phenylalanine in here? So I'm looking at my Mountain Dew. I mean, it's perfect, right? It's just sugar. Okay, no phenylalanine in my Mountain Dew, but a lot of labels will indicate if something has phenylalanine in it to help people with their diet. Okay. I don't know if anybody's vegan or vegetarian, but you can often, a lot of labels now will indicate whether something has agar in it, or that or agar or um, pectin, because the agar comes from pond scum, so it's technically a plant, so it's okay. The pectin can be derived from like the hooves of pigs, I believe, or something like that. So basically, they both have the same thing where they're kind of a solidifying agent. But because it is an animal product, a lot of vegans I know will not eat things that contain pectin. Okay. Anyway, PKU, not a fun disease. A lot of these diseases are not fun. A lot of these diseases just lead to mental deficits. It's amazing that we develop the minds that we have, right? I would say, well, maybe this explains why a lot of people vote the way they do. All right, no politics in there, because they're all wrong. All right. Anyway, um, we can also look at our glucogenic um, proteins as well too. So in this case, we have a few amino acids that can be degraded into pyruvate. Well, by a few, I should say several, because you can see ultimately our tryptophan, so tryptophan is both ketogenic and glucogenic, because it releases that alanine. And in alanine, if we transfer off the amino group, we can convert that into pyruvate. So basically, alanine and alpha ketoglutarate can interact to make glutamate and pyruvate. Okay, and we can I believe that's reversible as well too. But we certainly talk about alanine a lot with our lactate in the muscles there too. I believe that's our Cori cycle. If we look at cysteine, all right, I like cysteine. It's basically two steps. We're going to take away the sulfhydryl group. We're going to replace the amino group with oxygen, and boom, we got pyruvate. So cysteine, nice and simple. Only two steps in there. Yay! Okay. But if you look at threonine, we can cleave threonine to get our pyruvate through several steps in here, but we end up making serine as an intermediate as well too. It's kind of nice because with serine, our serine dehydratase. Now this is kind of funny. I don't know that this enzyme is, because we also are cleaving off the amino group there as well too, but we're removing water, so that's where the dehydratase part comes from. But we're also adding water to hydrolyze and get rid of the amino group that's up there as well too to make pyruvate. So that's a fairly nice little interaction that's going on there, but for some reason they call that serine dehydratase. If we look at glycine, so glycine is of course our simplest amino acid, but we have this wonderful little complex that comes in here. Oh my, yeah. So glycine, we're going to end up making this N5N10 methylene, which can then interact with our what they're going on there. Yeah. So we basically have a couple different pathways that we can have here. I mean, we can basically convert this into carbon dioxide in an amino group, and then adding one of these methyl groups to our tetrahydrofolate here, and then we can use that tetrahydrofolate to and break down glycine into carbon dioxide and amino group, and then bring this back and attach this here to make a 3-carbon amino acid, or 3-carbon compound, well, amino acid into serine. So we're essentially adding that to our alpha carbon there. Okay. Notice that we have pyridoxal phosphate as a cofactor in this. Again, we are transferring a single methyl group in there. So pyridoxal phosphate becomes critical for that. But this is certainly a nice confusing little interchange here. I don't know if you've ever been to it. It's a figure 8 interchange as opposed to like one of those roundabouts. When they first put a roundabout in and people don't know what they're doing and they stop and stop instead of yielding or something like that. Here we have some sort of crazy figure 8 interchange between glycine and serine. crazy going on here. Yeah. So, but anyway, just being able to follow this, follow these pathways and kind of understand what's going on. Um, what we're going through. I'm not asking you guys to memorize any of this. In fact, I could probably speed on through this, but I want to kind of go slow just so that we get practice following these pathways, because these are catabolic pathways. In the next section, we're going to be looking at anabolic pathways, and those can be pretty crazy. All right. So going back to our glycine, glycine has a couple different pathways in there. The main two, we can hydroxylate it to serine, okay, which then will go on to make pyruvate. That's what we saw in, this pathway here, going down this way. And we can also cleave it directly and produce carbon dioxide and ammonia in there. This seems to be a pretty big pathway in mammals. So we definitely want to control our ammonia. We're going to see that go into the liver and kidneys, of course. And we take our methylene group that's in there and put that on tetrahydrofolate. And then we kind of have this interplay between pathway 1 and pathway 2, where the one of the products of pathway 1 gives us the tetrahydrofolate that can accept the methylene group from pathway 2 and go back into pathway 1. And so we can see that regeneration in there. There's also a third pathway, okay, but this is a relatively minor pathway. So this involves an enzyme D-amino oxidase. All right, you're like, wait a minute, we don't have many D-amino acids, but wait a minute, glycine is achiral anyway, so it can fit in there, and it gets oxidized to oxalic acid to oxalate. And then oxalate can then accumulate and be a major component of the kidney stones in there. Okay, so if you have way too much glycine, you may see pathway 3 upregulated, and maybe that's a reason why some people have a tendency to accumulate kidney stones. It could be that they maybe just express a higher concentration of the enzymes involved in pathway number 3. I'm not sure about that, but that would make an interesting thought question, maybe a part of the take-home question there as well too. It's like, hey, speculate on kidney stone formation injury. No, that's maybe too much. But I like that fun question here. But again, the product you get there is this methyl glyoxal. Okie dokie, that's glycine, right? Okay. So those were compounds that went to pyruvate ultimately. We can look at ones that go to alpha ketoglutarate. Okay, so here we're looking at arginine, histidine, glutamate, glutamine, and proline. Okay, I like arginine because arginine, well, if only because of the one professor when I was in grad school who everything dealt with arginine. Okay, but arginine gets converted into ornithine, and ornithine is another kind of fun little amino acid that doesn't get incorporated into proteins, but it plays a lot of roles. I think we saw ornithine as one of our cofactors in one of our pathways, right? Hopefully that rings a bell. And it has some other actions in there as well too. But it's just kind of a neat amino acid. It's like, why didn't this one get incorporated? It's our design. Okay. But anyway, we can break this down in here ultimately. And you can see that glutamate, because it's kind of our central role, our glutamate to alpha ketoglutarate. We can see that amino transferase that's involved in there. So we can convert alpha ketoglutarate into glutamate if we need to go that way. But for our breakdown, we convert proline, arginine, histidine, and glutamine. All of them go to glutamate first, and then we get rid of the methyl group on there to make our alpha ketoglutarate, which we can feed back into any of several different cycles. Okay. All right. Recall that I mentioned that glutamate and glutamine make a good ammonia storage. So we have these amino groups in here. Here we've got 2 amino groups on glutamine. We can take one off to make glutamate. We can have some amino transferase to make more glutamine. So if we have a high protein diet, we might see a temporary storage of this excess ammonia in the form of glutamine circulating in the body, okay, to ultimately break it down, and it goes through the urea cycle in there. Okay, and again, our arginine degradation, part of our urea cycle, that's where our ornithine comes in. So interconnected. Everything is interconnected. I'm going to say proline. Remember, proline is our weird amino acid because we have this ring structure that connects to the alpha carbon. So we end up, doing this kind of half oxygen oxidase in here. So we get this kind of funky little reaction so that we can, activate our group here as delta 1 pyrroline, which makes it very easy to react and break this ring structure so that we can make this glutamate semialdehyde and can ultimately convert that to glutamate. Not that you need to know any of that. It's just kind of a fun little way to get to glutamate. I don't know, fun. I don't know. Maybe fun is too strong of a word, right? Okay. All right. So let's look at branched chain amino acids. Okay, this would actually be a great multiple choice question. I think I've asked this once or twice in the past, but I'm not sure if But it's something here like our branched chain ones, leucine, isoleucine, and valine. They don't get broken down in the liver. They get oxidized for fuel in the muscles, the kidneys, the adipose tissue in the brain. Okay, so that's just kind of an I would say almost trivia in there. But the fact that we have these little branched carbons in here, you might think, well, maybe there's a way we could activate this to cleave off one of those methyl groups and use tetrahydrofolate or biotin or something. No, they just we just have a specific branch chain aminotransferase that focuses on getting rid of the amino group first. Okay, so depending on whether you have valine, isoleucine, or leucine, in all cases we're removing the amino group from the alpha carbon and creating a ketone there. Okay, so we have these basically alpha keto acids. Okay, so replacing our ammonia with an oxygen. Then we have this branched chain alpha keto acid dehydrogenase complex. Oh yeah, it's a fun thing, but all three of these can bind to that. And you can see we can produce our acyl-CoA derivatives. So again, we're adding coenzyme A to these, okay? And we're cleaving off a carbon here, all right? So we have this nice little carbocation here. It's good for attack. And we can cleave off our CO2 there. We'll end up using NAD, making NADH from this, I believe, as one of our cofactors, and adding coenzyme A to get these acyl-CoA derivatives. Okay. However, if there is an issue with this particular enzyme complex, you end up with maple syrup urine disease. So this is where, like babies when they're born, if they have this particular defect in this enzyme, their urine will smell like maple syrup. So I would like to make a joke and say this disease was of course discovered in Canada, ha 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 ha, but I'm not sure if that's true or not. I want to make sure my jokes have truth in them. And again, maple syrup urine disease results in accumulation of these keto acids, and again you can see neurological effects from this. So a diet low in branched chain amino acids is critical to treating that. Okay. All right. So again, this is also important because this does not happen in the liver. I think we have the wrong thing here. Okay. If we look at valine, threonine, isoleucine, methionine. So methionine doesn't really have a branch in there, but it's part of this pathway. Let me see what's underneath. There we go. Okay. But these components can all make a succinyl-CoA. All right, so our isoleucine, no problem there. We ultimately end up making this propionyl-CoA, which can get to methylmalonyl-CoA, which we will see more of because we're going to use this as a component for building things as well too. But that can be converted into succinyl-CoA and feed into the citric acid cycle. Okay, so threonine, valine, isoleucine, and then methionine does this as well too. We can see this homocysteine that's up here as well too. So homocysteine, will play some important roles later on. And again, you can see that pyridoxal phosphate (PLP) is in some of these pathways as well. So we have a lot of cofactors. You can see vitamin B12, coenzyme B12, depending on how you want to call that in there. All sorts of good stuff in like that. It's really kind of neat to look at this because I had a friend who had some health issues that couldn't quite figure out. Every now and then there'd be like a racing heartbeat and some fatigue and some other sorts of stuff in there. Maybe she's a very pale person, so the pallor would be one of them, but that was one that was not noticed because she is very, very pale. But turns out is vitamin B12 deficiency. So here you can see one of the reasons that coenzyme vitamin B12 is important to help for this breakdown to make our succinyl-CoA and break down these glucogenic amino acids. And of course, the treatment is given more B12. Right? Now they're actually working on whether or not is our body not absorbing B12 from diet very well. So they're doing injections until they figure that out. But of all the things that it could be, tumors and all sorts of crazy diseases, and it's like this is like, okay, this is a very good relief. Right. But it just shows that these cofactors are critically important. We talked about how a lot of these, if we don't, if there's an error in these pathways somewhere, you can see many neurological effects, including neurological development being stunted. And this is why a lot of vitamins are thiamine and riboflavin and various prenatal vitamins are so important for the development of the fetus. So if you eat a really balanced diet, it's good. Prenatal vitamins also supplement, and that's good as well too. Oops, come back here. Okay. All right. So we've been talking a lot about nitrogen control within the body, but what about sulfur? Okay, so we want to conserve our sulfur oxidation as well too. So if we look at our nice little sulfur group here, our methionine breakdown, we can transfer this sulfur to serine to give us cysteine. Okay, so here we have our nice little homocysteine and our first step breakdown where we cleave off the methyl group in here. Notice that simple removal of the methyl group takes multiple steps, but we end up with this homocysteine, and then we can actually have serine come in and accept the methyl group here and end up making cysteine, and then we have this alpha ketobutyrate. That's just an intermediate in there. But the neat thing is we can use cysteine as our kind of repository for sulfur. Okay, and we're also releasing ammonia in there, but never mind. Too much. Okay. Finally, we can talk about, well, I won't say, yeah, I guess finally in this regards, we can look at asparagine and aspartate, which we haven't talked about too much. They can both be converted into oxaloacetate. And it's kind of neat because the first thing that happens if we take asparagine, we remove our ammonia group in there, our ammonia group over here, replace it with an oxygen, and we make aspartate, right? And of course, aspartate can get converted into glutamate fairly easily and leave behind oxaloacetate. Okay. So that's not too bad. Simple steps here, right? All right. So let's look at some disease states in here. Breaking down these amino acids is important for control. We don't want to accumulate all of these. Okay. So there are so many amino acid enzymes that there are tons of targets for these. Okay, so if you look at albinism, right? So I don't know if anybody's ever known anybody who was an albino, but there are I wonder if I can move this down here. I can. Good. Okay. But generally that doesn't seem like too bad of a disease. Okay, because it's just a lack of pigmentation. You end up with like white hair and very very pale skin. I say pink skin here, but it looks often almost white. The most dangerous part is in the eye, and it typically have the red eyes, so there's no protection against UV light in there. So most people who have albinism have to make sure they wear eye protection all the time so that they don't get UV damage to their corneas in there. But generally, of the amino acid defects in there, that might be one of the best ones because it's just like, all right, I'm prone to sunburns and maybe vision damage. Okay, not too bad. But again, if we think of albinism as a lack of melanin, well, melanin relies on tyrosine as a precursor. So if we can't break down that tyrosine, okay, then we can't make the melanin, so we can't get the pigmentation. So that's why you see that white hair, very pale skin. And you may have seen like albino bunnies. Like, every now and then, I come from a state where hunting is big, and they'll talk about somebody up in Canada was showing a picture. There's an albino moose out in their backyard. all the squirrels on campus. I haven't seen any albino squirrels on campus, but I know that there are some somewhere in the greater Forsyth County area. Someone had spotted an albino squirrel. So this happens. It's just, this nice little defect in there. But albinism is probably the most common. Well, second most common. These we're looking at per 100,000 births in here, maybe 3 out of 100,000 people have. If I jump all the way to the bottom, our phenylketonuria is the most common at 8 per 100,000 births. So I mean, it's not super common, but that is common enough to be noted on here. And we saw that we basically can't convert phenylalanine to tyrosine. So as opposed to not being able to convert tyrosine to melanin, it's like, oh, that's not too bad. Not being able to convert phenylalanine to tyrosine, you end up with this accumulation of phenylalanine in those phenolactate and phenylpyruvate that are building up there too. For some reason, newborn babies will not be able to keep down food, so they'll just constantly be throwing up, and often have a significant cognitive issues as well with PKU. Again, you can treat this by just limiting phenylalanine in the diet, and that is an effective treatment. It is very effective. Like I say, you can be pretty much have a normal lifespan once you figure out that you've got this disease. Okay? If you look at that alkaptonuria, we had our tyrosiniemias, but our alkaptonuria, fairly rare. We're talking at like 4 per million births. Again, we can't break down tyrosine. Okay, and particularly we can't break down that one particular intermediate in there. You end up with this dark pigment in the urine, and often arthritis. I don't know if you've ever met teenagers who have arthritis, but I've met some young people. It's like arthritis. You just think hey, Granny's got the arthritis, but no, and it's just basically this accumulation here. So this is not that bad. I guess arthritis is not fun, but it's a lot better than constant convulsions, vomiting, and cognitive deficit. Okay, all the rest of these you can see here. The very common things in here: mental retardation, a lot of vomiting, because you just can't handle the stuff that's in the diet in there. So your stomach is trying to. You get to see a lot of convulsions in there as well too. That's got to tie in with the basically this accumulation of these various kind of compounds in here. Not good. Your body wants to get rid of them one way or the other, and if it can't, then you see those convulsions in there. Our maple syrup urine disease, not a good thing at all. Fortunately, again, very rare. Vomiting, convulsions, mental retardation, and early death. It's like, yeah, hey, fun stuff. Same way here, our acidemia for our methylmalonic acidemia. So even if it's not directly one of the amino acids, here we can make our propionyl-CoA, but if we have an issue with our methylmalonyl-CoA mutase that converts that to succinyl-CoA, you get some pretty serious effects in there as well too. And again, this propionyl-CoA to succinyl-CoA, of major importance to the body. So you can see this all sorts of stuff in there. All right. We're going crazy talking about the diseases, but we'll talk more about amino acid diseases on Wednesday. Okay. As we're getting close to the end here, of course, every time I move something, it's up there. We saw in those pathways there are a lot of cofactors that are involved because a lot of times we are moving around single carbon units or small components in here. Okay, so let's talk about a couple of these. We're going to talk primarily about tetrahydrofolate, something called S-adenosylmethionine or ADOMET. And if we put the S in there for some, we call it SADOMET because you had to be kind of a sadomasochist to want to study this, right? Okay. And then biotin. All right. So biotin, if we look at that, we saw this earlier. It transfers carbon dioxide in here. Biotin, again, we have this nice, lovely, long little valeric finger that kind of sticks off of there and has our CO2 group on it. So this nice little tether can move the CO2 group to where it needs to be. Okay. Oops. Put the wrong thing here. Tetrahydrofolate is really versatile. Okay, so we've got this methylpterin base in here. We have this nice little aminobenzoate in the middle, then we have a glutamate on the end. So we have lots of areas where we have potential reactive groups. So we can do a lot with this. You can see we have either this benzene ring. We have a series of conjugated double bonds here. So there's a lot of particular reactivity. Tetrahydrofolate is an essential vitamin. Again, you got to have low tetrahydrofolate, low folic acid, bad things. But the key here is that tetrahydrofolate can transfer one carbon in a variety of different oxidation states. So here we have, our methyl group. Here we have a alcohol group, and then here we have our aldehyde group. So 3 different oxidation states, but we're still transferring this single carbon. So because it's so versatile, we see tetrahydrofolate used in a very large number of metabolic reactions. It's an amazing cofactor. Again, that's probably why it's critical for the diet in there. Generally, we're going to get these carbons coming from serine. Now, lots of things can get converted into serine, as we've seen, but we're going to use our serine often to bind to this in there, and then we can interconvert the different forms on the tetrahydrofolate before we see its usage. Okay, so our H4 folate, I forget which are 1, 2, 3, 4. I think those are the 4 H's that give it that, but we can oftentimes, maybe we don't have H4 folate, but H2 or H1 or H3 or something like that. Anyway, so in this case, we're looking at that ring structure in there. So if I go back up one, right? Yeah, we're looking at our ring structure in here. We can see these nitrogens are going to be the ones that are often involved. Okay, so here we can see our most reduced, our methyl group can often be attached to that in there. Here we can see, our alcohol group in there, which can actually interact with the other nitrogen from that central aminobenzoate section in there. But our tetrahydrofolate, we can make N5 methyl tetrahydrofolate, N5 N10 methylene tetrahydrofolate. We can make this N10 formal tetrahydrofolate. We can make all these different intermediates in here, depending on what we interact with, whether it's serine or formic acid or something like that. But the key is tetrahydrofolate has a lot of ways that it can stabilize single carbon molecules, so it can transfer them in transfer these single carbon molecules in a variety of oxidation states. Okay. All right. So that's why we see it used a lot. Okay, let's compare tetrahydrofolate with our adomet. So our adenosine group in here, we're basically reacting adenosine with the methionine, and then our nice little methyl group that's down here attached to the sulfur, because now we have this additional interaction in here. It makes this methyl group really good to transfer. And in fact, the methyl group on an adomet is 1,000 times more reactive than on the tetrahydrofolate. So tetrahydrofolate is more versatile. It can do a lot more stuff. So that's why we see it used a lot. But our adomet can do one thing really, really well: transfer that methyl group super well. Okay, and basically helps regenerate N5 methyl tetrahydrofolate in here, which is our only known use of that in mammals in there. But our adomet is really good at transferring a methyl group. Okay, so let's try and summarize this. The previous chapters we might have had like 1 or 2 bullet points to summarize stuff. Well, here you go. Bam! This is where it really, really starts to hit. Okay, so looking ahead, this next section is an ordeal, which is where we're gonna play games or try and have a little more fun with it. Okay, and I'll be changing up. Exam number 3 will probably be completely take home, because this next section is just beefy. All right. So, but this chapter, here we are. We got our amino acids. They're an important energy source. Okay, so we think about eat your meat so you build strong muscles. Yeah, of course we can use our amino acids that we consume to make other proteins in our body, but we can also use them for energy. Okay, so the first thing we got to do is transfer the amino groups off of them to give us glutamate, because we need to control the nitrogen in our bodies in there. All right, so. And when we generate this ammonia, we want to quickly recapture that and make carbamoyl phosphate. So thinking CPS1, for our critical enzyme into the urea cycle. Amino acids can be degraded into pyruvate, acetyl-CoA, alpha-ketoglutarate, succinyl-CoA, and or oxaloacetate. Or, so as we can see here, would be our ketogenic, and then the rest of these would be our glucogenic as well. Okay, so if we directly get acetyl-CoA out of them, those are ketogenic amino acids, and then the rest are glucogenic because they can make our glucose. All right. There are a number of diseases that are caused by defects in the various enzymes of the various pathways in amino acid catabolism. And then we have a ton of cofactors that are involved in here as well too. Okay. So it's kind of neat. I've looked at my old exams that I've given in the past, and I've looked at some of the information I have on here, and I'm like, oh, I can't believe I didn't ask this. It's like, so it might be a good idea to know what THF, Atomet, biotin, pyridoxal phosphate, all those fun things do, and say, maybe question, well, which one is best at transferring a methyl group? You'd say, oh, Atomet, because it's that activated sulfur group in there. That methyl group will just kick off really easily. And which one's most versatile? You could say tetrahydrofolate, or all sorts of stuff. What do they all have in common? Kind of a big long tether region in there. Tetrahydrofolate less so, I guess. But anyway, tons of information in there, right? I know I'm getting to the end of it because I'm starting to ramble a little bit. Woo! Ugh. Okay. So let me stop the sharing here and ask you guys questions, comments, apoplexy.
I had a question.
Yes.
So the assignment about the disease, there wasn't a place to submit that online.
Okay. I will make that available. I know we'll be talking about that. Okay. No, did I make that due today?
Yes.
Oh, okay. Yep. I will make that. I will open up a submission for that. Yes. Yeah, that's right. I did make the fatty acid oxidation one due today. Ooh, I forgot about that. Yeah. So I'll open it up for you guys. I know I saw at least one or two answers to the citric acid cycle one. There is. Yeah. Okay. But before I get to those assignments, anything about this material first? It's a lot, right? Yeah. And it's kind of sussing out. That's why, and like I say, on Wednesday, we will go over exam stuff too. So what did I do here? All right. Well, I've done something funky. Here we go. Okay. Yes. There we go. That's what I needed to do. But so let's take a look at some of the other stuff that's out here.
I had one more question.
Yes. Yeah.
So all of the animations and everything, since I don't have Apple, it's not letting me look at them.
Yep.
I'm just looking like up, animations on the web, but is there anything like specific that those animations would have that I wouldn't find like on Khan Academy or something like that?
Right. No. I generally try to stay away from Khan Academy because I didn't want to make this a comment, but Khan Academy often has better ones in there too. I've been meaning to go through those. Yeah, actually I can't, it's not even an Apple thing because it's just Chrome is a Chrome or Flash, Flash has just kind of disabled those. I was kind of looking at a few and I've forgotten there's so many distractions, so much stuff to do. But I should just kind of like unpublish all those until I can find replacements for them. I know there's like one or two that still work because they're not Flash based, but yeah, don't sweat those too much. Those are all just kind of like little extra things in there too. And it's a shame because there are a couple that I really liked, but certainly for this next section, I'm already peeking ahead at some different animations for you guys that'll hopefully that are maybe Unity based or something like that that are not Flash based. So don't sweat those too much. Those were just extras. I don't know. I liked them, but yeah, technology changes, right? Okay. So if you haven't already created a citric acid cycle question, make sure you get that into me today. Okay, you still have another 13, almost 13 hours. Yeah. The other thing I wanted to pull up today, yes, okay, so let me share screen one more time. Um, I'm not sure if this is, yeah, actually, let me look and see if it's in here first for you guys. Um, all right, I have a citric acid cycle worksheet for you all. And this here. Ah, it's not, okay. Um, I Well, that's under. Oh, okay. Hold a minute. I will have to move some stuff around because it's basically another assignment. But let me share the screen and I'll show you what we're looking at here. I think this will actually help you prep for the studying for the Krebs cycle, for the citric acid cycle, TCA cycle, whatever you want to call it. But there's a nice little 2-page worksheet here. Well, actually, it's really 1 page, but the second page has the answers. Awesome, right? Okay, and I think, had I made this available, this also might have made it easier to make your TCA cycle questions in here. But um, I think this is very helpful because it not only asks you for the name of the compounds that are in there. Um, does it talk about the enzymes? I don't think it does talk about the enzymes. But one of the things I like about it is you can see that, okay, here we are, we're cleaving off, a carbon, we're making a 2-carbon unit that's going to combine with our 4-carbon unit. So here's our oxaloacetate and our pyruvate that's getting converted to acetyl CoA. We lose the coenzyme A. We get these 3. We get our citrate and our aconitase and all our good stuff in here. And you can kind of go around and see that. Okay, well here we have 6 carbons. Oh, we're going to lose one of those as carbon dioxide to make a 5-carbon unit. Here you can see we're going to lose another carbon dioxide to make a 4-carbon unit. We got this stuff going on here. Yay. So, it kind of, shows you all this fun stuff here. Okay. All right. And then, like I say, the answers are on the next page here. So this is a good practice filler in here. So,
Is that one already uploaded?
um, it will be. I have it uploaded, but I don't have it published because I have it in an assignment folder, and I don't want to make this an assignment for you guys. So I'm going to put it under the exam 2 material, and then it will be available like right after class. Okay. All right. All right. So let me stop sharing there. Okay. And then the other thing I want to do, put this up here. Okay. Yeah. I really need like 3 screens. Okay. Exam 2 study guide. All right. Okay, so the last thing I wanted to kind of share with you today was the study guide that's coming up. Because, like I say, our exam is going to be on next Monday, most likely. So there's a fair amount of stuff in here. Okay, so this is a longer study guide. Okay, um, and I'm still by Wednesday I will have the exam pretty much finalized, but a lot of it is going to be on the same stuff there too. Like clearly you're going to have to know the TCA cycle. It's like draw the TCA cycle from memory. It won't quite be that bad, but I want you to know everything that happens in the TCA cycle. The good news is I know there are some bonus points that are available on this as well too, because everything in the TCA cycle, there's a lot. So there may be a few things that are extra. If you get everything, you get bonus points. I guess if 25 of the 30 things that are involved in the TCA side, I don't know. Never mind. I'm getting far ahead there too. But it becomes kind of critical here. How can you divide this down? And I know some people have made like little flashcards of each step that's in there. What's the enzyme involved? What is the reactant in the product? Is this regulated anyway? Are there any things that come off or come in to the reaction? So whatever kind of works for you. So so definitely know the TCA cycle. I know there are a few where you have like some multi steps in there. I'm not going to ask you for each little breakdown of how that reaction works. Like step 1, step 2, step 3. No, I just want you to know the enzyme and what we start with and what we end up with. So the 8 major components of the TCA cycle. And I think that worksheet that I'm going to make available here in just a few minutes will help you with that. Um, so other than that, the TCA cycle is almost the entirety of chapter 16. There's a few other things in there too. We talked about pyruvate dehydrogenase. So, or our pyruvate dehydrogenase complex, sometimes called PDH, sometimes called PDC. So this is our trifunctional enzyme. It's got 3 separate reactions using 5 cofactors. So be able to talk about pyruvate dehydrogenase in some detail. The main thing is it's going to convert pyruvate into acetyl CoA to feed into the TCA cycle. So it's our lead in there, but that trifunctional enzyme, it's like, what does part 1 do? What does, and what do the different parts do in there? Know where our carbons are coming from or going to. Okay, yeah. And then anaploidic reactions, being able to tell me, define an anaploidic reaction and what that really means in there. Okay, so chapter 16 is not that bad. It's just memorization stuff. Okay, our fatty acid catabolism, a little more, chapter 17 is a little more, less memorization, more application in there. So we can talk about, what's the advantage of fats, where does fatty acid oxidation take place, as opposed to fatty acid synthesis. So fatty acid oxidation, of course, is in our mitochondria. We can talk a little bit about breaking those fatty. Of course, we have our triacylglycerols. So we have glycerol and we have our free fatty acids. What happens with our free fatty acids, and what happens with our glycerol in there as well too. For our beta oxidation, we can look at all 4 steps that are involved in there. So we got that trifunctional protein. We can look at saturated fats, monounsaturated fats, polyunsaturated fats, and odd numbered carbon chains. Okay, so not a whole lot of craziness going on in there. And then we end up with the ketone bodies. What are the ketone bodies and where and how are they used? Okay. So that's fairly straightforward. And I think chapter 17 is the most normal of the chapters. Chapter 18 with our oxidation in here, we have our various proteins. Where does, where do the proteins for the energy come from? So a lot of it comes from our diet. And of course, we have our breakdown of proteins in our body, whether we're, doing muscle damage or just natural turnover or whatever that's in there. Our first step, what's the first step? We want to control our nitrogen balance. So we got to look at that deamination there. Usually the first thing that's going to happen is we're going to take off that amino group. And where does that go? Into urea or uric acid or ammonia, depending on the organism. But we're mainly going to talk about the urea cycle. So I want you to tell me about pyridoxal phosphate. Should probably put in the abbreviation for that, PLP. Look at our pyruvate alanine cycle. Carbamoyl phosphate and CPS1, that critical enzyme for our entry into the urea cycle. All that sort of that good stuff that's in there as well too. All this stuff that we talked about at the end of today as well. So chapter 18 is a little more, because we just have so many amino acids. We're looking at 20 amino acids that get broken down, right? And then they often have these multi-step breakdowns. So what you'll likely see is on the in-class portion, it won't be that bad, and I think it's pretty straightforward, other than, trying to memorize the entire TCA cycle, but, defining an anaploidic reaction or, talking about these different beta oxidation pathways, depending on what free fatty acid is in there, or, defining what a ketone body is and how it gets utilized, and, any advantages or disadvantages of that. And our different components of the amino acid oxidation. I'm not going to ask you to memorize any of those pathways, just maybe, ketogenic versus glucogenic. Um, but yeah, it's the in-class portion I don't think will be that bad. The take-home portion will probably say, well, speculate. All right, so how would you treat this disease? What causes the disease, or something like that. There's a couple I found in the past that I'm like, oh, this is fun. This is fun. So, but it just becomes kind of applying what you take from these 3 chapters and looking at what happens if we accumulate a particular component in here, or what does one of these intermediates end up doing in the body, maybe something like that. And for example, if we look at those diseases of the urea cycle, you can see that a large number of those diseases resulted in vomiting, convulsions, and mental retardation. So it's just like the metabolites in there are clearly involved. in neural development and regulation, as well as something with to do with the digestion. So maybe not digestion, could be part of the convulsive reaction there too. But yeah, so there's some commonality in there too, and it's the weird ones like albinism or alkaptonuria that don't cause things like that. Right? It's like arthritis instead. That's the only one that causes arthritis. Anyway, yeah, that's what we've got there. So I know I actually talked a little bit longer than I expected to about that, but what I want to pull up is that one other assignment. What I need to do is put all these in one nice little place. Oops, that's the wrong one. Supplements. There we go. So our fatty acid oxidation case study. Ooh, can't show you my answers in there too. Okay. But yeah, I will open the Dropbox for this as well too. And this shouldn't be too awful because I don't know how you guys are in terms of health. If you ever Google your symptoms, and it's like, oh no, and clearly it's a brain tumor, right? But this is one of these things where you get experience kind of Googling symptoms and then kind of weeding out the places like, oh, clearly you need to eat more kale. No, but you can find out these diseases and kind of try and figure out what's going on here. Okay. So I just kind of want to see what you guys can do with this. I'm hoping that you can get everything exactly right. Now it'd be awesome, but really the main focus of the grading is going to see how are you troubleshooting this? How can you figure this out? And this is kind of like a test run for a question you might see, probably not in this much detail for the take home portion. Okay. Yeah, you can see I got a bunch of questions in here, right? Yeah. Is this one of the, yeah, okay, and this is the, yeah, right, it's true. What concerns might you have about marrying someone from the Faroe Islands if you're a carrier for SPCD? Oh my Lord. Okay, yeah, see? Crazy. All right, okay. All right. It's like Dr. Ebert hates the Faroe Islands. No, that's not it at all. Actually, I think I hate it. Aren't they, I guess, is that the Argentinian name? Falklands, Faroe? I can't remember. Anyway, the whole goal is to try and have a little bit of fun with this and kind of see if you can figure this out. All right. So I know anybody who's thinking of medical type career, this should be like a fun puzzle. All right. Fun puzzle. I don't know. Yeah. Because all fun puzzles have terribly ill children, right? All right. I was going to say, yeah. Yeah. I also like this question here of, should you worry about your child having this disease, or how common is this disease? It's an awesome answer. All right. Anyway, I didn't actually share that, did I? I don't know if I did or not. Anyway, but play with that. I'll make that that open up there. Anybody have any more questions or anything? All right. No. I'll see you in person on Wednesday, so that will be good. And then we'll go over more of these case studies, this urea cycle case study and this fatty acid oxidation case study, and kind of play around with those and talk about the answers and then talk more about the exam. Okay.